Hello, doctors. Welcome back to our 90-day University Prof Pre-Final Mastery. So here today we are going to start our general microbiology topic. We'll be having three general microbiology topic, which is easy because we have came all the way through systemic microbiology. So this general will be very easy and definitely very important for exams also. So let's start our topic. So before going to the topic, you know what we do. What is this 90-day University Prof Pre-Final Mastery means? You know that we are having most important things: rapid revision, volatile facts, quick revision, high yield topics of all subjects, and most expected topics will be discovered. Yeah, that's the main thing here. Now, what else important here? You know very well that our one second. Yeah. So here, if you see today's questions, the topic which we are going to discuss here, we have long notes and short notes. First will be the long notes one. In long notes, we are going to discuss about, see, the most important one, bacterial cell wall. Okay, bacterial cell wall. Sorry. So here what we're going to discuss would be the bacterial cell wall. So bacterial cell wall structures we're going to discuss, and difference between gram-positive and gram-negative bacteria. Then the sterilization itself is a big question, and then going to be a bacterial growth curve, very important question, all expected question. Culture media, sure, 100% guaranteed questions will come from this topic. I can guarantee you. Okay, and maybe two or three also can come. Everything is important here, and that's what Auricle has choose. Very good. Their question bank is excellent. Okay, you can expect all the questions. In Wick reaction, gram staining again, Cox postulate very easy, drug resistance and plasmids. Okay, so these are the things we will be covering today. Now, so this one, why choose Oracle? You know, again, same thing. We will discuss again to say we have very good recorded classes. We have very good recorded classes, right? And then live classes going on, practical sessions you're going to have, exam module and hard copy set. These are very, I know we have it specially made for you. So those are things are going to be very, very helpful for you, of course. And then yeah. So recorded classes are going to be, very, when you talk about recorded classes, it's going to be flexible learning, expert instruction, and convenient revision. That's the main thing here. And then when you talk about other things, live classes, which is going on now, which we are doing now, it's very in-depth coverage, and also experienced educators going to give you the knowledge. Interactive learning will be there. Yes. Skipped few topics. So exam express live classes, and then we have this exam module, live classes, express exam module. We will tell you how to write the answers briefly, if the diagrams and pictures needed. And one-to-one doubt clearing session is available for some students who have taken that particular plan. They will also have this direct contact with us and explaining. Hard copy sets are available, good for writing important notes and everything. And it's a very, very good question bank. I would say this is the best for MBBS students because nothing they can come out of these topics. And yeah, so these are the things very important. Now these are the plans we have. See, when you see our subscription plan, this is a subscription plan. So we have a foundation course and then advanced learning course, the complete learning and ultimate MBBS plan. So look, whatever things are available, and you can use my code Dr. Santhosh Rajan 20 for a discount in the subscription. Okay, so these are the things that is going to make you better. Now, what we're going to do is we're going to start our topic here today. Yes. So the first question, the first long question here asked is, that's a beautiful question, very good question. They asked about the structure and the function of the structure and function of a bacterial cell wall and how different gram positive gram negative. So when they talk about it, you're going to write a bacterial cell wall first, bacterial cell wall. And then what are things here in bacterial cell wall? Okay. Now bacterial cell wall, if you see, you draw as just a bacteria. Okay. Just simply draw a bacteria like this, and then you mark. Okay. This is the one outside you're seeing. These are the flagellas, and you have pili. Okay. So these are the things, but here, this is your cell wall. You're going to write a cell wall here and cell wall, and then what you're going to write here, and then you can always write other diagrams, so nothing is wrong. Okay, when you draw the picture, good picture, they will have. So inside one, which you're seeing is your cell membrane, and this is your flagellas and pili, and then your nuclear material. Okay, nucleolar material is not a nucleus, but nucleolar material you have, material. And all the ribosomes and every structures you can add, whatever structures you know, you can add it, ribosomes and all other structures, whatever you know. Okay. Now, this is a cell wall. Now, cell wall differs in gram positive and gram negative. Before going to gram positive, we'll write the functions also. We'll talk about the function and then we'll go to the structure. It will be better. Now, when you talk about the functions, what are the function of cell wall? A cell wall is outside, no, it's a protective layer, barrier there, right? So first thing is protection. For protection, first thing is protection. Second thing is it helps in the movement because it's outside the cell. That means it helps in the movement of the substances, right? So one is protection, other one is the movement of the all the substance coming in inside. No, those movement. It helps in storage of granules. You remember storage of the anything, storage of the any molecules, whatever the important storage molecule or whatever. For storage, it's helping. Fourth one, you know that this is where the antibiotic acts on the cell wall. That means this is having a role in antibiotic resistance. It has a antibiotic resistance. It has a role. Okay, and that is this place. If you remember, example, beta lactam drugs, they act on cell wall only, so for that purpose only. So movement of all the molecules, protection, storage, these are the basic things. Apart from that, other things, if you want, you can maintain protection, again, maintaining the osmosis. Okay, protection, osmosis. So these are the important 5 functions you will be talking. Okay, that's all. Now we are coming to the structure. So structure, we actually classify the structure based on gram positive, gram negative. Okay, the structure, I'm going to write the structure. Structure is differing from the gram positive bacteria, gram negative bacteria. Okay, when you're writing, you write it fully, gram positive, gram negative. So we draw a picture. For gram positive, how you're going to draw a picture is that you're going to one big layer, one thick layer like this. Okay, you're going to write it as peptidoglycan layer. And here, if you are good in drawing, it's better to draw clearly so examiners will be so happy. There is this molecules, NAM, NAG molecule, okay, NAM, NAG molecule, and they all are connected by some bridge called pentaglycine bridge. The two molecules NAG, they are connected by pentavalent bridges. Okay, you just only mark it, pentavalent bridge. And you see this peptidoglycan is a very thick layer. Okay, thick layer. So this is a thick layer of peptidoglycan. And of course, below that, you will be having the cell membrane. One layer you're going to draw, that would be cell membrane. Cell membrane. That's all. Okay. Apart from that, important thing is that you're going to draw some teichoic acid. Okay. Teichoic acid is very catalytic for gram positive. But when you come for this one, here you're going to do a little bit less. See, the peptidoglycan, you're going to make it a small layer. Instead, you're going to add some other layer, which is very thick. That is your, just simply some picture, LPS layer, lipopolysaccharide. Lipopolysaccharide layer. Okay, saccharide layer. And this is your peptidoglycan. Small layer. You see, peptidoglycan in gram negative is small. Peptidoglycan is very small. And of course, inside would be your cell membrane. There would be a cell membrane. Okay, cell membrane. So draw a short picture like this. Okay. And gram positive, so everything is positive here. When you talk about gram positive, everything is positive here, right? Everything is positive in gram. So this is gram positive. So what are the things you're going to write here is in gram positive. So first of all, they will be purple in color. Gram stain, it will be purple. And gram stain, it is purple color. Second one, they have peptidoglycan, peptidoglycan layer, thick, very thick layer. And here it is thin, okay? And here it's grams purple, here it is pink color, pink or somewhat red color, okay? Pink color. Think this will be thin and thick. And other one is important here is this pentaglycine bridge. You have it only in the gram positive. Pentaglycine bridge. It's positive only in here, but here it is negative, okay? And then important is that you're going to see teichoic acid, teichoic acid, okay? Teichoic acid. These are the things. Here you don't have it. No, we don't have it. Instead, what we have important layer, lipopolysaccharide. Lipopolysaccharide layer. Lipopolysaccharide, which is the one which is producing, if you remember, endotoxins. Endotoxins. That's all. Okay. So these are the things important when you're writing a structure of gram positive and gram negative bacteria. Okay. Gram-negative bacteria. So these are the things you have to mention. So now you already have all the things are described here. If you go, everything's explained. We have slides also. You can go through the slides. It's already there with previous classes. Say we have everything. So you can go through this one. cell certain function. The picture is given better here. You can go through this. Okay. So even for TB also it's mentioned mycolic acid liporaminomenin is present. Okay. That's all. These are the things you should know, right? Now, next one is your now, methods of sterilization. The next question is methods of sterilization and their application in microbiology. Okay, it's a good question, definitely important question. So when you talk about the sterilization, we usually talk different types. One is a physical method and chemical method are the two common methods, right? First of all, sterilization means itself, you should know it is killing all organisms, bacteria, virus, everything, fungi, parasite, all parasite with spores. That word is important. Which spores we are killing. Okay. Then comes your classification. So when you're going to put a classification, you are going to say there are two types. So what are the two types? You're going to say it is a physical method and the chemical method. Physical and chemical. Physical and chemical are the two important methods. Now, in physical method, you're going to write a physical method and classify it. You're going to put it like this. You're going to put two things like this. One is your dry heat. You're using a dry heat. If you draw this picture itself, the examiners will be so happy. They will know that you know the answer. Other one is your moist heat. Moist heat. Dry heat and moist heat. Okay, dry heat and moist heat. So when you talk about the dry heat, what are things you should know? You should know this is a flaming. Number one, flaming. Number two, red heat. Number three, incineration. Incineration. And other one is your autoclave. Your hot air oven. Hot air oven. Okay, these are the things. Now you have to explain each thing. So when you talk about flaming, this is for test tubes. In whatever the test tube you're using in the lab to sterilize it. Okay, test tube, mouth, for the test tube sterilization. Test tube, mouth sterilization you're using. Red it is basically for the loops and wires. For loops and wires sterilization in the lab, whichever the loops and wires you're using, for that we are using. Okay. And incineration is basically to burn the biomedical waste, yellow bag. All the yellow bag waste, we are using this one. Okay. Hot air oven, we will talk in a while. Hot air oven can be a short note also. Okay. So we want to talk about it. In moist heat, we'll talk about the moist heat. We'll go next page. Okay. Okay. Now in hot air oven, the first one we're talking about hot air oven. In hot air oven, temperature is very, very important. You know that. So in hot air oven, what is the temperature? It's usually 160 degrees Celsius for 1 hour. Okay. Shortly remember 1 hour or 2 hours, 1 or 2 hours you're using. So what are the substances you're using? So here in hot air oven, all the glasswares and the surgical instruments, surgical sharp instrument. Okay, and then what else? Any your dusting powder, gloves dusting powder is there. No, all the dusting powder in the gloves, not the gloves, the dusting powder of the gloves you're using. Okay, and liquid paraffin. Liquid paraffin. Okay. And you have to always write a control. Always control for hot air oven and for everything. So biological control, which is the one which you're using, which bacteria we are using here? We are using hot. So hot tea, remember like that. Okay. Clostridium tetani, commonly used Clostridium, Bacillus subtilis. Bacillus subtilis. These are the two biological controls you're using. Spores of these two, tetani and subtilis. Even globogy you can write, but this two is more than enough. Okay, right. Now for the moist heat. Now the next one is your moist heat visit. So in moist heat again, you're going to write all the things very nicely. So first one, you mention it as less than 100 degrees Celsius and then equal to 100 degrees Celsius and more than 100 degrees Celsius. So you list it like this, and in less than 100 degrees Celsius, very commonly, that is your pasteurization. Pasteurization. Other one is your inspisation, right? And equal to 100 degree, you will be talking about which one you are tyndallization. Okay. Yes. And above 100, you have only autoclave. Very important one, autoclave. Okay. It could be a 5-mark question also. You can get in 5-mark question. So when you talk about the pasteurization, we have two methods, holder and the flash. Holder and flash. We talk about holder method. So a common standard method. So it is 63 degrees Celsius for 30 minutes. Everything about 3 and 6s. See, 3 and 6s. Okay. Flash is like fast. So it is 72 degree for 20 seconds. You have 2. So 20 seconds. Short. It's in a flash of moment, right? That's all. What about inspiration? We talk about inspiration. You have to write. It is 80 to 85 degrees Celsius. Okay. Temperature. 80 degree temperature. 3 consecutive days. 3 consecutive days. Okay. What about internalization? This is 100 degrees Celsius at 100 degree. This also 3 consecutive days. Consecutive days. Got it? That's all. Simple. Then, what is the use? These two, they're used for like serum media like LSS media and sugar containing media. Any sugar containing media, sterilization. That's all. Okay. You have to write this. Now autoclave. Autoclave in this, you have to draw the picture first. Any autoclave, you write vertical or horizontal water, but at least one should be there to show them what are the structures available. Okay, I'm writing in a red, you use other color. Okay, here. Okay, so here is inlet, outlet, pressure gauge, temperature. There is this body. Okay, and then this is the body. Then here you're keeping all the sterilizing substances, surgical instruments. Okay, so you're going to mention it as a pressure. You have a pressure gauge. This is a lid basically. It's a lid, pressure gauge, temperature monitor. There will be a temperature monitor also. Okay, temperature monitor. And pressure also. This is the body jacket. It's a body we call jacket, a jacket proof layer. And then here the surgical instruments are kept. Okay, you draw this picture and then temperature you're going to put how much? 121 degrees Celsius for 15 minutes and 15 pressure. At pressure 15 kilo, that is pascals. Okay, pressure. This is the standard temperature. Now what is the control we are using here? The control for this is a steam, no? Moist heat is a steam heat. So you're using Bacillus stearothermo Bacillus stearothermophilus is the control. That's all. This is the one we are using. Okay, for moisture. That's all. This based only you're going to do this application in the microbiology. Very, very simple. Okay, yes. But then last one now we're going to do is the chemical method. In chemical method, at least few things you have to mention and write it. First of all, the alcohol. Okay, alcohol, and then aldehyde, and then your chlorine compound, then iodine compound. Okay, chlorine, iodine, and one more thing is that you have to add your ethylene oxide. Ethylene oxide. This can be one more questions also, but also this one. So when you talk about the alcohol, you know that we're talking about two alcohols. Isopropyl alcohol 70%, ethanol also 70%. This one specially for thermometer. Thermometer we can use thermometer. And both can be used for hand rub. Okay, hand rub. Sanitizer. Aldehyde, again, we have two aldehydes. One is three actually in case, if you remember. One is your formaldehyde, 40%. Glutaraldehyde. Formaldehyde. Glutaraldehyde is 2%. You know, very important question is that. And then orthophthalmide. Orthophthaldehyde. Okay, orthophthaldehyde. Okay, now what are the uses? Now, first one, formaldehyde anatomical specimen we are cutting, and also for your fumigation. We use it for the fumigation also, fumigation purpose. Glutaraldehyde, you know, for endoscopes, so important endoscopes. Orthophthaldehyde is basically same, it's a glutaraldehyde, but no need any activation. That's all. No need any activation. That's it. Okay. Chlorine, very important. It's for swimming pools and drinking water. Okay, swimming pool, drinking water. You can use common. One thing, and most important is a hypochlorite solution. Don't forget your hypochlorite. hypochlorite, 1% hypochlorite solution. This is used basically for any spills. Any spills in the hospital, we use sodium 1% hypochlorite. Iodine, you know, that is the tincture iodine. Tincture iodine. One of the wound disinfectant we can use. Okay. Ethylene oxide, you know, this is one very important for the respirators in ICU, respirator sterilization, and also for your heart lung machine. That's all. This much only. Okay. This much if you write, it's more than enough. You'll get 10 out of 10. They can easily give you 8 to 9 marks. Okay. They have to give. Okay. Now, so next one, we finish the sterilization. So you can see all the, you can see her. Yes, so everything is mentioned. Dry heat, all the things we have spoke here, it's given here. And then pasteurization, inspection, moist state we have already. And then autoclave, everything is given here. Okay, everything is mentioned here clearly. And then now chemical sterilization, most of them are covered. Okay, that's all. Now bacterial growth, a very easy question. 10 mark means you shouldn't make any mistake in this one. Okay, so now whenever they say bacterial growth curve, what you're going to, first thing you're going to do is draw the picture. First you have to draw the picture. Okay, how you going to draw it? Draw like this. So your growth curve, it will be starting like this. First phase, this one, this, and this. Okay, so what are the phases you know? So the phases are, first one is lag phase, log, stationary, decline or death phase, involution or decline phase. Okay, so these are the phases: lag phase, log phase, stationary. So now, first of all, in the lag phase, you have to say something, and then you have to write number one lag phase. Number two is your log, or we call it exponential phase. Okay, and then your third one is your stationary phase. Last one is your decline phase. Okay, now look at this. So lag phase is basically after vacation. It's, you can say, after vacation. So what happens in lag phase? The bacteria will go maximum size. Maximum size. The bacteria goes into maximum size. Okay, bacteria goes maximum size. Exponential phase, now they're active. So now the bacteria start to divide. Okay, division of bacteria happens. So division of bacteria. So when division of bacteria happens, automatically what happens? Size decreases. Size decreases. Cell size decreases, but multiplication of bacteria happens, size decreases. So this is the phase exponential growth happening. So you can do all the important things here. What you can do? You can put your biochemical tests are done here. Biochemical tests, the staining methods, all the staining, even your antibiotic susceptibility also you can do here. Antibiotic susceptibility testing. Everything can be done in log exponential phase. This is also 1 mark question they can ask. Okay. Stationary phase, we already have yes. So spore forming is here. And T is there. T for basically toxin. We are talking about exotoxin. Yeah, exotoxin. Exotoxin. There is A also here. A. So A for also bacteria also produces antibiotics. Okay. It will also produce antibiotics. And of course, bacteriocins. Bacteriocins. These all are produced in stationary phase. But in stationary phase, what happened? The number of live cells will be equal to dead cells. That's what it is stationary. Okay. Number of live bacteria is equal to number of dead bacteria. In decline phase, dead cells start to decrease. Bacteria start to die. Bacteria start to die here. Die. So count decreases. The bacterial count decreases. This much only. Growth curve means you have to mention only the detailed explanation. The points by points you write, that's more than enough. Okay, so usually it's 10 mark, but usually 5 mark only they'll ask. Don't worry, they'll not ask for 10 mark. This will go in 5 mark question only. Okay, right. Next. Okay, so now so it's given here. You can see all the lag phase, everything is mentioned very clearly. So you can see this picture and go with this. Okay, that's all. Now, next question is your describe various culture medias. This is another important question. Can be short note also, culture medias and their uses in the cultivation of the microbes. When we talk about culture media for growing the bacteria. So what are the culture media is important, you know. So first of all, we have to classify them. Culture medias can be simple media, number one. Number two would be enriched medias. We have enriched media. And then we have enrichment media. Then we have also transport media. Then we have differential media. The last one is your selective media. Okay, now what are the things you have to know here? So simple media is basically the basic media. Simple media, they have the basic constituents. They have the basic constituents. Okay, so for this example, you're going to give is your, here the classic examples which you're going to give is, example is peptone water, nutrient agar. They are basic ones, nutrient agar. Okay, that's all. Enriched media is a media where the media is enriched. Okay, you're enriching with substances. Which substances? You're enriching with the substances like could be a blood or could be a serum or could be egg. That's all. Okay. So when you talk about this, which are the examples you're going to say? When you say the blood, this is blood agar. You're talking about the blood agar. When you talk about serum, you're talking about low flux serum slope. Low flux serum slope. That is for your diphtheria. Even blood agar and chocolate agar also. Here, chocolate agar. Agar. Okay. Egg is the LJ media. Lowenstein Jensen we're using for TB. TB, this is for diphtheria. Corynebacterium diphtheria. Okay. That's all. This is enriched media. Enrichment media definition is basically here in enriched media, it basically supports pathogen, inhibits the contaminants, commensals, which we don't need. Those are inhibited. Okay, suppose the pathogen inhibits the commensal. So here, important examples you can give as enrichment. For cholera, you can give for Vibrio cholera, also for Salmonella shigella, Salmonella shigella. So for Vibrio cholera, we use alkaline peptone water. For shigella, this you can give your enrichment. You would add selenite half broth. Selenite half broth. One example. Even tetrathionate, one is enough. Selenite half broth is enough enrichment. Okay. And transport media. Transport media is to just maintain the viability. Here only maintain the viability, not multiplication. Maintain the viability. The bacteria should be alive. That's it. Okay. For this, you have again important examples. If we use Stroud's or Emmys. Stroud's or Emmys media for Neisseria gonorrhea. Okay, Neisseria gonorrhea. Whereas also you can use VK media, Venkatraman and VR media, Venkatraman Ramakrishnan media, Carrie Blair media for again Vibrio cholera. That's all. Example. Differential to based on your differentiating two properties. That's all. Okay. So here you can say your example should be McConkey agar. McConkey agar, clad media. Example. Okay. Selective media, selective for only that particular organism. It is selective for one type of bacteria, selective for one type of bacteria only. Okay. So for this, many examples are there. Example, if you see, one second. We have cetrimide agar for your Pseudomonas, right? And then you have your example, you have TCBS media for Vibrio cholera. For Vibrio, we have TCBS media. Okay. And also, if you remember, there is this DCA Salmonella, Shigella, Salmonella agar for Salmonella, Shigella. One of the examples. That's all. Okay, so these are the few examples for selective media. Just more than enough. Okay, I've combined it, wrote it. The explanation is here. You can go through these notes, of course. So this is more than enough. For example, will be happy. But yeah, you can elaborate and write a little bit more if you want. You can add some pictures. You see, simple, enriched media, everything you have given. Okay, now culture media, okay, everything is given. Enrichment, transport media, selective media, so many examples. You choose at least two examples is more than enough if you give for each bacteria. But for 1 mark purpose, yeah, you have to know all the important medias, selective medias you should know. That's all. So these are the things here. Anaerobic, I forgot to tell about anaerobic. Anaerobic, only thing is that Robertson cooked meat media. You will write about Robertson cooked meat media. Okay, it's mentioned here. That's all. Okay, fine. Now, in-week reaction. Now we're coming to short notes. Short notes are very easy here. Here, basically, short notes. In-week reaction means you have tried what is in-week means. I for indole. I for indole test. And M for methyl red. VI is basically works Worksprushness. Okay, there is, I think there's a little bit spelling once again. Proscure. Okay, it's basically proscure once again. Proscure. Okay, proscure. You can see from the spelling from behind. Okay, it's very difficult. It's this names or no some other. Works proscure. Okay, one second. Okay, so we are here now. Works Prushkar. Okay, Works Prushkar reaction. Okay, Works Prushkar reaction. And then C is for citrate. C stands for citrate. Now, what happens is that these tests are basically to differentiate some of the bacteria in instant moment. Okay. So, indole test, you basically look for a ring. In methyl red, it will be red in color. Works push also it go red in color. Citrate means that it has to become a blue color. Okay, change into blue color. That's okay. That's if you don't write also doesn't matter. But here, for Escherichia coli and Klebsiella, you have to write. So MVIC is basically IMVIC, right? You're going to write like this. So now what happened for Escherichia coli, this test would be indole will be positive. This one methyl red will be positive, but VI will be negative. But for Klebsiella, what happened? These two will be negative, but VI and then citrate will be positive. Was pushed and got be negative. That's all. This is the MVIC reaction, the short method for the biochemical reactions. Okay. Indole ring, methyl red, Vogt-Prushker, and citrate test. So this is the one thing you want to write. In-week reactions basically biochemical tests, 4 biochemical tests to identify, to differentiate few bacteria. That's all. Okay, that's much only. Nothing much you're going to talk there. Okay. Yes. Now next. So we have mentioned it here. Very important. Proscolia. Now gram staining versus ZN staining. Okay, both are differential staining. First of all, both in differential, both are differential staining. Both are differential staining. Now, we're going to differentiate both now. How we're going to differentiate? First, I'm going to write here the gram stain. On the other hand, we are going to write ZN stain, Zeil-Nielsen staining. Okay, Zeil-Nielsen staining. Zeil-Nielsen staining. Okay. Otherwise also called as, or it's also called as acid fast staining. That is also important question. Acid fast staining. Okay, this you're going to write. And then what is the use of these tests? This one, gram staining, it will differentiate the bacteria to gram positive and gram negative bacteria. Zn acid for stain mainly we're using for mycobacterium species. Okay, mycobacterium tuberculosis or leprae, doesn't matter. For you're using for mycobacterium species. Okay, right. Now steps. What are the steps here? For gram stain, the first step is, okay, remember like violet, Miss Violet, so crystal violet. It's a primary stain. The next step is violet intakes, indole. Okay, indole is a fixative. Oh, sorry, iodine. Oh, 1 second. Okay, second step here is you're going to iodine fixative. The next step after iodine is sure, you're going to add the acetone decolorizer. It's a decolorizer. The last step is safranin. Saffron is a secondary stain. That's all. Okay. That's all. Gram positive, gram negative, differentiate gram positive and gram negative bacteria. So the gram positive bacteria would look how? Purple color. The gram negative will look pink or red color. Pink is better named, or red doesn't matter. It almost looks like pink only. So here what happened in microbial tuberculosis step, what are the stains we are using? We are using first of all carbol fusion. Then we are doing heating intermittently. Heat intermittent. Then you're using 25% H2SO4, sulfuric acid. It is sulfuric acid. And the end, we are going to use methylene blue. Methylene blue. Okay. So here what happened? You will be seeing the bacilli is, you will be seeing what color? You will be seeing acid first. You will see red color bacilli. Beaded appearance. We'll show the picture now. Okay, red bacilli beaded appearance you will see. Okay, now you go to the next slide. Clear? So this is the way how this is the gram positive bacteria. How does it looks? It's a gram positive. How it looks? Like see purple color here, and you see down this the pink color one is a gram negative, and these are bacteria gram positive. Not important for this question, right? Just for your notes you've written. I think Zn is not mentioned here, but it's okay. Whatever I told you previous the slide that you remember. Okay, that's all. Okay. Now next one. Cox postulate. When talking about the Cox postulates, you remember this is five postulates. Five postulates. Cox said, what is that? He told that every every lesion is every you can remember every organism is involved with any lesion. Okay, I mean it's not every is a wrong word to use here. Yes, there is always an organism. What he said was, he said what? Always there is an organism involved in any lesion. First of all, organism involved in a lesion. And now from that lesion, you can isolate the organism. You isolate the organism. Basically, in culture media, you're isolating the organism. Okay, right. For third step, the same organism, whatever you isolated, this organism, if you inoculate in lab animal, they have to produce lesion again. Okay, this organism which you isolated, if you put it in the lab animal, they have to produce the lesion. Now again, this lesion you must be able to isolate in a culture. You must be able to isolate the same organism in culture. Very simple. That's all. Okay. This is the four. Fifth one is basically molecular. Means all organism have genes that causes the disease. They have the gene that causes the disease. Very simple. Okay. So an organism comes, if you confusing, remember an organism, some bacteria, it is causing some lesion. It is causing some, let's say, lung lesion. Now from that lung lesion, you start again. From lung lesion, you're putting on a culture plate. You're growing. The bacteria is isolated. So your organism is isolated. Okay. Now that organism again you're taking from there and you're putting in a lab animal. Let's say a mouse or something. You're putting in the lab animal. So you get what? Again, you get the lesion. You got the lesion. Now that again you take that lesion, it looks like confusing, but it's very easy. You put what? Again you grow it, then you will have the culture. The organism is again re-isolated, same organism. That's all. Okay. This is the very simple Koch's postulate ka, the meaning. Okay. You will know it very easily, not much difficult. Okay. Yeah. So it's already mentioned here also. You can go through it. Koch's postulate. Okay. All the 5 postulates are given here. You can go through it. Drug resistance. When we talk about the drug resistance, this could be actually bigger because I think we did it in genetics part, in immunology part. Yeah, it's okay. We will do again, no problem. When we talk about drug resistance, we have 2. One is the vertical, other one is horizontal drug resistant. Horizontal drug resistant transfer. Okay, resistance, the drug resistance transfer. Okay, so vertical means the resistance is passing from mother to the babies, whatever you call mother to child bacteria. We're talking about, it's not a child, it should be a mother to daughter bacteria or whatever. Okay, mother to child bacteria, that's called this thing. Usually this vertical. In horizontal we have many things. In horizontal we have something called transformation and transduction. We have conjugation, transformation, transduction, conjugation, and the last one is lysogenic conversion. Okay, transformation, transduction, conjugation, lysogenic conversion. So when you talk about the transformation, free, any free any free resistant gene or what, okay, any free gene or genetic material from environment is taken inside and the bacteria becomes resistant. Okay, so it's a bacteria. You take this genetic material goes inside, and then it causes a drug resistance is transformed like that. Or transduction means where a bacteriophage is involved. Okay, a bacteriophage is involved in causing the transduction. Okay. Conjugation means one bacteria to another by using something called conjugation tube, which is called sex pili. Okay, we call it a sex pili. One thing. Lysogenic conversion basically when the lytic, we call it as the non-lysogenic form, which is not dangerous, they become lytic, they become virulent because of some phage, again some phage. That's all. These are the mechanism where how to spread it. Okay, so now you can go in the detail because we're getting short of time now. You can everything is explained in this thing. You can go there. One second. Yeah. So this is the method very clearly mentioned, transvertical or horizontally. Then again, each thing, conjugation, transformation, we don't have much time to say. So you go through this. We have explained it in other topic we have explained. So it's okay, fine. If you just go through it, it will be there. Okay, transduction, lysogenic conversion, and the examples are given very clearly. Okay, go through this much only, not much. It's easy. You will know this one. Okay, next comes on. Plasmids and what they are important microbiology. Okay, this is also very important topic. You see here, when I talk about the plasmids, it's basically an extra chromosomal material, right? It is basically an extra chromosomal material. Usually, if it combines with the host double-stranded DNA, circular DNA, it's extra chromosomal material, it exists cytoplasm. So the plasmid mixed with the host chromosomal DNA integrates and they form something called episomes. They form different types of plasmids we have. What are the plasmids we have? We have Hef plasmid, R plasmid, Col plasmid. Hef plasmid is something which forms the fertility, so sex pili is formed. R means resistance, which forms the resistant to antibiotics. And Col is basically which code for the bacteriocins. It's code for the bacteriocins. Okay, so there's colicin, pyocin, diphtheriocin. And there's a virulent plasmid that is for toxins. It causes the toxins. That's all. These are the different type of plasmids. You should know. Okay, that's all. So we're done with this plasmids now. Yeah. So once again. Mm. So that's all. So we're done for today. So once again, sorry, I took the last slide. I'm sorry. Yeah. So everything is clear. I know that you must have, it must be you'll go through it, you'll understand it. It's not that difficult, but yes, all important questions we covered in this topic. So you have to be thorough with that. Okay. So once again, thank you for watching. For Oracle, all the best. Do well. We'll catch up in the general micro biology next class, class 2 soon. Okay. That's all. Thank you.